Morning. You're lucky because Friday afternoon, Frederick said he couldn't make it because he's on clinical rotations, getting his MD and PhD. And I emailed another one of our speakers, and she's still in Africa, and she couldn't make it by today. And you were almost going to be bored by me talking about energy. So we're actually very lucky that didn't happen, and he was able to get the morning off from his doctor on clinical rotations. And where are you in Frederick?
I'm sorry?
Where is your rotation?
Oh, so I'm in geriatrics right now, actually.
Oh, you're right place.
Yeah. So I'm in Haven Hospice.
Frederick is a PhD and MD candidate. He defended his dissertation in May. Now he's on clinical rotations. He's going to talk to us about a useful, harmless virus, adeno-associated virus, to cure genetic diseases, and how his work is used genetic sequencing to improve the tools we have. And so as an engineer, I've told him he has to explain it to me because I'm not an MD, PhD candidate. So Frederick, Ashby, thank you for coming today.
All right, well, thank you for having me. I'll try not to bore you guys with genetics. And I promise you guys it's not really as complicated as the title implies. So a little bit about, hold on, this isn't working. I think the clicker's not working. Yeah, the clicker doesn't work. All right, that's better. A lot more information on the other slides. So a little bit about the area that our lab works on specifically. So gene therapy is a very broad area, just like in mechanical engineering and electrical engineering and all that. You can have all kinds of subspecialties and very specific projects. So what we worked in is lysosomal storage disease, which is a genetic disease that you get when you're young. And these diseases are kind of rare, but not really. It's one of every 5,000. So you might have met someone who has one of these. Some of them, they can range in severity from being essentially asymptomatic to being fatal within 2 years of birth. So you don't have to worry about all that. Essentially, there are these garbage cans in your cells called lysosomes, and they dispose of all of the junk that accumulates in your cells over time. And when those are dysfunctional from birth, it can cause all kinds of issues. So essentially, every single system in the body is affected by lysosomal storage disorders if you have one of them. Here's a picture of someone with Sanfilippo syndrome. You can see there's very marked differences in the facial bone structure and everything. So it's very noticeable and very detrimental to parents and patients as well. So this is just a bit more about the consequences of having lysosomal dysfunction. It can be kind of what they call a positive feedback loop. So if you have a wrench thrown in the system, you can imagine it just tears things up and makes things worse and worse. And over time, you wind up having really bad inflammation, really bad what are called oxidative species, which is just something else that really causes a lot of problems on a cellular level, which leads to all these symptoms that you see with lysosomal storage disease. Sanfilippo syndrome type B is a very rare lysosomal storage disorder. And for this reason, it's kind of neglected. And that's part of the reason why our lab worked on it. So within the lysosomal storage disease umbrella, there are mucopolysaccharidosis, which is a category. And within mucopolysaccharidosis, abbreviated as MPS, there's MPS 1, 2, 3, and so on and so forth. So subcategory 3 is known as Sanfilippo syndrome. The purpose of this is just kind of illustrate how subcategorized it is and how detailed each of these diseases can be. And you will need a different gene therapy for each of these diseases. So as you can imagine, there's a lot of work to be done in the area of medicine and biology to take care of a lot of these kids. So we work on type B specifically. As I mentioned, it's characterized by very early symptoms from birth. There are very aggressive neurodegenerations. Sometimes the parents call this child onset Alzheimer's, colloquially. The symptoms are very simple, and unfortunately, they generally do become kind of unresponsive by the time they reach around 10 or 12. First age of onset for disease is 2 to 4. However, it's important to note that the average age of diagnosis is closer to 8. So a lot of doctors don't really know how to diagnose it. It can't really be diagnosed clinically. You kind of need to do genetic tests. So the doctor may be going through a lot more common diseases first and trying to rule those out. And so they can get bounced around from doctor to doctor. Your local community private practice family physician is likely not going to have any idea how to diagnose that. So they're probably going to refer you to an academic institution who may refer you to someone else. So that's part of why there's such a delay there. Symptoms include, hyperactivity, developmental delay, and all kinds of organ failures. Notably bone dysplasia, which I'll talk about a little bit later. You don't have to worry about that. There's a lot of different types of Sanfilippo syndrome. There's not just one specific mutation that causes it. These mutations for Sanfilippo syndrome can happen anywhere on the genome. And actually, majority of people with Sanfilippo syndrome, they kind of have happened or arisen spontaneously. Although there are some populations that are more likely to have it than others. So essentially, this can kind of be approximated to something like IQ a little bit. So this is developmental quotient. This is gray matter. This is DQ, which is similar to IQ. So you have an advancement up to a certain point until they get to around the age where they start degenerating. And then you can see it just kind of plateaus to the point where they become catatonic. So it's kind of terrible to tell, you imagine telling parents this, that there's this disease, this is what's going to happen, there's nothing you can do. So that's part of why, from an ethical standpoint, I kind of pursued this for a few years and tried to see if we could advance the field a little bit and hopefully bring this closer to having some kind of therapy that we can give parents and patients so that we can't just diagnose this as a terminal illness at the age of 8, right? So AAV, for those that don't know, the gentleman who was kind enough to introduce me did his homework and noted that AAV is a harmless virus. So essentially adeno-associated virus. It's not related to adenovirus at all. It was discovered as contamination in the 60s. It's essentially a dependovirus, which means that it relies on other viruses in order to even replicate. So because it can't replicate on its own, it really doesn't cause any known human illnesses. It's not completely harmless. I mean, if there's anything that you inject directly into your body, if you don't do it carefully under the correct conditions, you can hurt someone. However, in this case, for gene therapy, we do that in such a way that it's controlled and we can achieve favorable outcomes. So essentially, for those who don't know, viruses, they go towards your cell and they inject DNA. And a typical virus injects DNA that causes it to make more viruses. So AAV, like I said, is not able to do that independently. So what we do is we actually take out all of the pieces of the virus that encode for making more AAV, take all that out, and we replace it with a therapeutic gene. So essentially, these are just empty virus capsids that deliver a therapeutic gene and achieve favorable outcomes. And that sounds great. Does it actually work? Well, it actually does. So these are all of the FDA-approved therapies that are already out, meaning not only has this gone through clinical trial, this is actually available to patients, these ones right here. So this is for SMA1, which is another childhood onset disease, which has really favorable outcomes, actually. But the long-term studies, it's important to note that we need to do long-term studies to really see how long this works. So the idea is you have these patients who are missing or they have a defective gene, and you just give them an AAV infusion with whatever gene they're missing. And as long as the AAV is able to reach whatever target organ is mostly affected by that disease, they should have at least partial resolution of their symptoms. It works really great for eye stuff. The reason why is because your eye doesn't really grow, if that makes sense. So for that reason, for AAV specifically, it makes a lot of sense to treat eye disorders. But it also hits the liver very well. The problem with the liver, in the case of AAV, for those that don't have background in this, is that the liver tends to turn over a lot. What that means is that the cells in your liver tend to grow and die off a lot faster than other organs. So for that reason, AAV can kind of taper off in terms of its overall effect, but it's debatable of how how much that effect actually causes a loss of treatment. So that's the case for hemophilia B. Hemophilia B is a bleeding disorder. And in this case, they use AAV to target the liver and create the missing clotting factor here. So anyways, that's a little bit about AAV. This is just a little quick literature review I did a few years ago in my early dissertation, just to kind of characterize how many trials are actually being done on Sanfilippo syndrome type B, how many are AAV focused and that kind of stuff. So actually, there's not even that many that are dealing with intervention. So you can see all of the clinical trials registered in the United States, only 7 are actually focused on interventions, and 4 of them are focused on what are called enzyme replacement therapy. So remember that I said that there's a defective gene in the case of Sanfilippo syndrome and most of these other genetic disorders. Genes encode for enzymes. So the logic here is you just recombinantly create the protein or the enzyme, and you just inject it into patients. It sounds great in theory, except for most of these enzyme replacement therapies, specifically for Sanfilippo syndrome type B, have not actually worked very well. So you can inject the enzyme, but it doesn't distribute to where it needs to go quite as well. 2 of these trials are dealing with AAV, which would be an ideal candidate for targeting the brain and other structures in patients with Sanfilippo syndrome. However, both of these, again, this is kind of just background knowledge, both of these AAV trials are using what are called wild-type capsids, meaning these are capsids from viruses. They're just essentially directly taken from nature. We haven't modified them, we haven't done them better. So from an engineering perspective, you can find all kinds of structures in nature that work out great from a structural standpoint, but they're not necessarily made for our specific use case. So from an engineering standpoint, we can take these wild-type viruses and we can actually improve upon them so that they can hit whatever target tissue we're trying to hit better. Because in nature, the virus doesn't really care about efficiency and things like that. But we care because we are trying to infuse this for the purpose of curing someone. So this is one of the first parts where we used barcoding technology in order to try to improve the speed at which we can screen our engineered capsids. We tested technically 21, but for the purposes of this, it's 19 different engineered capsids that we wanted to test simultaneously in the same animal. We do this through barcoding technology, and I'll get into that a little bit later. So a major bottleneck in trying to progress in the field of AAV is it's so expensive to run clinical trials and also animal trials. So there's a lot of protections in place to ensure that there's ethical use of animals. There's problems with using animals just because we are not the same thing as a mouse, right? So there's a lot of overlap. And mice are really cheap, but at the same time, what happens in a mouse does not necessarily translate to a human, right? So we work with what we have, and we try to optimize our workflow so that we can get to clinical trials with safe and effective treatment faster. And that's the idea here. So we have a lot of different engineered capsids. These are all different capsids of AAVs that were tested simultaneously. Essentially, each of these were created independently. We made a vector mix of all of these different capsids. So we have like an equal amount of this, equal amount of this, equal amount of this. And each of these different capsids that we have in their DNA, they have a tiny little barcode that tells us, the researchers, what this actually is, which capsid it is. And so what we do with this, the advantage is we can essentially just inject one animal and have that animal act as a control group for all of these different AAV capsids simultaneously. So we don't have to inject 256 mice, we only have to inject 12 mice. So with that, this is just a little workflow you guys don't have to worry about. So we extracted DNA from the organs of these mice after being injected using some different methods. And we can actually get a lot of data on, just to give you an idea, all the different body parts, where each of these different types of AAV went to, and which capsids are really good at hitting specific organs. And we can get data on both DNA and RNA. So if you guys are familiar with the central dogma of biology, we all have DNA, That DNA gets transcribed into RNA, and the RNA gets turned into our enzyme, our protein, which in this case is what's defective. So it's important for us to know not only where is the virus going, which is the DNA level, we need to know where it's actually getting transcribed into RNA as well, which we can get from this. So this gives us, again, a lot of data to go through, and we can see broad level sort of distribution characteristics. This is to be expected. Most of the AAV that was injected goes to the liver and spleen. Interestingly, not a whole lot stayed in the brain, but out of the viruses that expressed the RNA, the functional virus, most of it stayed in the brain where we injected it. So essentially, what we were able to find is that we can learn a lot with a very small amount of mice if we're just smart about how we have our reporter genes. And in this case, we just used, like I said, barcoding technology to ensure that we can tell where the AAV went and where it's transcribing without actually having to do any sort of complicated or expensive assays. This is just more information, just giving an idea as to where overall all of the AAV went when we injected it through various methods. And from that, we can do some fancy statistics to ask some big picture questions, which is a bit more useful than just going through all of these complicated graphs, right? There's a lot of debate going on right now as to the utility of mice. I'm not sure if you guys keep up with the FDA news, but apparently the FDA relatively recently reduced the restriction on requiring animal trials before going to human subjects. So that sounds great on paper. I think it's a net good thing, but at the same time, majority of law-abiding, good conscious scientists are not going to want to move on to expensive non-human primate studies and are not going to want to go straight to humans without at least doing something in animals just to make sure there's no toxicology issues or something like that. Because like it or not, we are very similar to animals on a biological standpoint. So if something is going to go wrong, we want it to go wrong in the mice first. And if it doesn't go wrong in the mice and there's something else that'll go wrong in primates, we want it to go wrong in the monkeys and not the people. So that's the principle. We start with mice because, just for lack of a better way of putting it, they are cheap. And it's very easy to get a big clinical cohort of mice. It's a lot easier to do that than it is for monkeys. And for monkeys, it's, like I said, way better to test on those than humans. So if there's not really a correlation between monkeys and mice and macaques or monkeys, then why are we even using the mice in the first place? So in AAV, this is again just from the background of AAV, which you guys wouldn't be expected to know, but there have been a lot of engineered vectors that in AAV that have worked great in mice, but they work terrible when they go to monkey studies. So then they question, what is the point of using mice if it doesn't correlate? So I went back and I checked, and although there may be some isolated scenarios where things work in the mice but then don't work in the monkeys, overall on average, there's a strong correlation with the stuff that works well in the mice compared to what works well in the macaques, if that makes sense in this one. So essentially there's a difference between a perfect correlation and a loose correlation. For those that may not be mathematically inclined, that's okay. So essentially you can have, a formula that kind of tells you, if you have X, you multiply and you add a certain amount, you will get Y. In biology, we don't get that so clear cut. Sometimes it's a loose correlation, but there's still a correlation. So what we essentially found through statistics is that things that do better in the mice are more likely to do better in the primates. And that's actually really important for the research community because it justifies us using mice for screening for our engineered capsids. Because mice are way cheaper than macaques right now. A lot of biotech companies are actually buying a lot of the monkey facilities just because they want to all test their vectors. So monkey studies are extremely expensive now. And for those of us poor academics who are relying on the NIH grant funding, which doesn't increase with inflation, by the way, it's important for us to be mindful of the taxpayer dollars. Okay, so this kind of goes into the same stuff that was in monkeys. So this is just kind of highlighting what I was talking about before. Generally, we try stuff in a dish, in human cells that are grown in a petri dish, and we kind of see if it works there. We try it in a cheap model, like some kind of rodent, because we like to be efficient. And if that works, we generally go to macaques, and we go to people if it works. However, obviously, these are not the same, and these are not the same. So famously, in the COVID pandemic, people remember how everyone was saying, there's no way they could have done this so fast. How did they do this so fast? Well, essentially, that's what happens when we look at researchers and we just say, money is not an object, do whatever you want, and do everything you can. So actually, with the COVID vaccines, they did everything that they would normally do on the scale, but they did it all simultaneously. So they did what's called parallelization, where they did the mouse studies, they did the macaque studies, and they also recruited some very early phase 1 clinical trial participants that were willing to participate and accept the risks. So because of that, they did essentially 10 years worth of work in 1 year, and they didn't lose any quality in the process, which is actually pretty ingenious. So that's what happens when we fund research. We tested 2 different injection methods, cisternal injection, and this is essentially when you inject directly into the brain itself. We did the same thing on the mice as well, and we wanted to see if there was a correlation. Essentially there was. It didn't matter if we injected into the brain directly or into the cerebral spinal fluid space. And going through all here, we saw a relatively similar distribution with some differences between how the mice responded versus the macaques. And so essentially I did some fancy statistics to ask some, again, big picture questions. So a lot of times in AAV research, we see things that are in triplicate. For some reason, science and maybe engineering too, they like triplicates. However, there's no statistical basis for a triplicate. It intuitively sounds good because if the 3 are similar to each other, you can presume that they're probably correct, but you don't know that. So with the data that we had here, I have a lot of information on the variation and dispersion of how the same capsid can respond in different animals. So with that, I was able to calculate how many monkeys do you need in order to have a statistically sound and well-powered study. And with that, the answer is actually not 3, it is around 6 or 7, which is, again, important to know for designing future trials. So this is probably only relevant for researchers, but if you see a study with AAV that's comparing different serotypes and it's less than 6 or 7, then it might be underpowered. Of course, this is only based on these capsids right here, and different capsids may behave differently, but it's just food for thought. And we saw general correlation between the central nervous system and the rest of the organ system, between the macaques and the mice, essentially. So we have more stats to validate that it makes sense to be using mice for screening and use macaques as a secondary. So another thing that we did essentially was we asked some broad questions, again, about the research field of AAV in regards to sex assigned at birth, biological sex. So in animal studies, it's been shown that different genders of some animals can actually respond to AAV differently. And this is not really known why. It's been speculated. So what we did with the RNA that we extracted from some of our macaques is we stratified them by their biological sex, and we wanted to see what genes are being upregulated and downregulated in their brains that would possibly explain why they are responding differently to AAV. This is a list of all the genes that wound up coming up. We looked at different sections of the brain, used some fancy bioinformatics tools, which essentially run computer algorithms to try and find correlations that would take forever for a person to kind of sift through. Most of the differences were on the X and Y chromosome, which is not surprising at all, considering that is one of the main differences between males and females genetically is the X and the Y chromosome. And this is just a Venn diagram just to kind of show a little bit of the different areas that they came up with certain genes. And there was some overlap. So these are different regions of the gene. We have the temporal lobe, cerebellum, and midbrain. We did that just to kind of get a good idea as to different geographical regions of the brain. And a lot of these in the middle here, these are actually Y-linked genes. So that just comes into the genetic sex differences between males and females. So case in point, most of the geographical regions of the brain had different genes involved. So each region of the brain may have a different mechanism for why there are differences in how they respond to gene therapy. And these were just kind of looking at different pathways that may be associated. Interestingly enough, some of them dealt with inflammation, some of them dealt with reactive oxidative species. So that's like starvation pathways and things like that. Interestingly, there were also some genes that came up that deal with COVID infection. So for those that don't know, males are actually more likely to have worse outcomes with COVID compared to females. And that's even after you account for everything else. You account for how healthy the male is, how old they are, and everything. For some reason, males are more vulnerable to COVID-19 than females. And it's been hypothesized that it's because of this gene called MAFB, and we actually found that come up. Another thing I got to do was going back to lysosomal storage disease specifically, look into the idea of using brain stem cells to regenerate some of the lost brain tissue in these patients. So obviously I didn't do it in people, but I did it in these mice that we use that have Sanfilippo syndrome type B. So essentially, we injected neural stem cells that were tagged, they glow red here. This is a mouse brain that we injected. These are the neural stem cells. Wherever we injected them, they didn't travel very far, so we wanted to explore this as a potential therapeutic. Essentially, we mostly found that when you do brain parenchymal injection, which is where we go here, here, and around here, that was one of the best methods for injecting these cells. This is just general methods. The key thing here is that I had issues with traditional fixation methods. So typically what we would do is we would take the brain, we would embed it in ice or wax or something, and we would actually chop it bit by bit. And then we would look at it under the microscope. But the problem is you would cut, and then look at it, cut, look at it. But we may be missing something in those areas that we are not seeing, if that makes any sense. I know it's not intuitive, but just for purposes of explaining the methodology here, what I elected to do instead was do what's called 3D immunostaining, where we actually took the brain, we fixed it, processed it, labeled it so that the red cells come out more red. And then we actually clarified it so the brain became transparent. And then we ran it through an imaging software, and it actually created a 3D image of the brain so that we could, in real time, take a look at where the cells actually went to. And from that, we can take virtual sections, and we can take a look at the distribution of the neural stem cells we injected. And so we can see that these two injection methods actually did the best. And intracerebral ventricular injection, which is where you go through the top of the brain, actually did really well, particularly in the, I forget the exact structure, I think it was the globus pallidus, but that actually had a profound amount of distribution. So if this were to be repeated, we may even elect to do a modified injection method, which involves both this method and this method at the same time. So that's essentially just figuring out what is the best way to inject these stem cells so that we can regenerate the most amount of brain tissue possible. That's very far off. And then this one was actually looking into outside of the brain. How can we account for if we're actually hitting our targets and if we're actually addressing things outside of the brain like bones? So I mentioned that the face bones of Sanfilippo syndrome type B, they have notable differences, but there's also long bone differences as well. So like femurs and stuff like that. So just like how folks can get a hip fracture, these folks with Sanfilippo syndrome, they're like 8 years old and they can get hip fractures actually. So it's very detrimental to them. So in our mouse model for Sanfilippo syndrome type B, we actually don't have a good way of measuring if we're fixing the bone issues of the disease. So this is what I sought out to do, is see if there actually are bone issues in our Sanfilippo syndrome mice. This is just a little bit about bone formation. It's not really that important, but essentially you have what's called epiphyseal closure after you're fully grown and your bone is done growing, done getting longer essentially. So in humans, that typically happens between 13 and 14 years, depending on your gender. And in mice, it happens around 5 months of age. So essentially, we took the femurs out of the mice. Obviously, they were dead at that point. And we ran them through a CT machine to actually quantify bone mineral density and bone mineral content and other morphological features of the femurs. It's analogous to a DEXA scan, but it's with a CT, so it's more powerful. So when we look at the center of the bone, right here, we can see that there are some differences in bone mineral content for both males and females. Interestingly, in the males, there was increased cortical thickness. Cortical is just this region right here. I won't go through all this one by one, but essentially there are some anatomical differences that are different, importantly, between females versus males. And that was the significant thing that was found there. And when you look at that over time, what I showed on the previous slide was just when the mice were 3 months old. Remember, epiphyseal closure happens at 5 months. So we have different time points where we're essentially looking at the progression over time. This is males and this is females. And you can see that these graphs are different. And that's the important key note, is that in Sanfilippo mice, it seems to matter whether you're a male mouse or a female mouse. And that's important for study design. It also potentially has implications for how we treat humans if this can be replicated in humans. And then we also look at the diaphyseal region. This is generally where your growth plate lives. And we take a look at that. And this actually showed more significant data than the midpoint in the diaphyseal region. But again, there's this disconnect between male and female mice. And I won't bore you and go through all of them individually. Essentially, again, like before, we see a difference between the male Sanfilippo syndrome mice versus the female Sanfilippo syndrome mice. The black is normal, and the gray is our diseased mouse. And same thing with density and content. We see a little bit of a divergence in that the males seem to become more attenuated over time, and the females actually become more pronounced in their differences over time. So that's all radiological data, but after working with a bone lab, I wanted to try to see if we can get some biomechanical data as well, just to kind of get an idea as to whether these are actually functionally different than normal mice. So the way this works is that you have a 3-point bend, point here, and a hydraulic press records this and presses down until it breaks. And then it also measures the force as well as the displacement. So that's just a picture of it. And then eventually it breaks and we get our data and we analyze it. So you can get all kinds of fun physics-related metrics from this. We can get stiffness, ultimate load, work to fracture, all that stuff, all just from breaking a bone. So relatively simple process that can give us some useful information for if this is a functional difference. And we actually did find differences in our Sanfilippo mice compared to the control mice. And again, we do have differences between the females versus the males. So the amount of work to fracture was markedly increased in the Sanfilippo syndrome mice. It should be noted that these femurs actually took longer to break, so that's probably the reason for this. And the ultimate load required to fracture these bones was also increased for both males and females. However, in the males, this kind of tapered off after around 6 months of age, whereas in the females, it really started to take off after 6 months of age. Reason for this, we don't have a clear explanation. But it's important to note for when you're designing animal model or animal trials, because there are a lot of researchers that might not necessarily keep track of whether the mice were male or female. So you can imagine if there was a research lab that's not tracking whether the mice are male or female, this can really throw off all your data if there are differences in the gender skew for each of the experimental groups. And that's the importance of this. Ultimate stress showed no difference, and stiffness only became pronounced in the females at around 12 months of age. And we showed a statistically significant correlation between all of our radiological data, as well as the ultimate load and the stiffness of the bones, which is just kind of validating that these radiological metrics can be used in place of doing these more involved biomechanical tests, generally speaking. This is kind of getting into what I mean by correlation. Does not always mean a perfect correlation. It's R squared of 2, which is definitely not great. Up here, it's quite a bit stronger when you look at ultimate load. But in biology, unfortunately, we don't get perfect correlated data all the time. There can be a lot of biological noise and a lot of genetic variation. Okay, so essentially, we are the first to characterize this in the Sanfilippo syndrome mouse. We noticed sexual dimorphisms in that males and females responded differently with the disease. And we noticed that these differences do translate to actual biomechanical differences. And we suggest that this mouse model can be used for determining whether treatment was effective in hitting the bone manifestations of Sanfilippo syndrome type B, because there are differences. And we think that PQCT or DEXA could possibly be considered for monitoring treatment correction of mucopolysaccharidosis disorders in the future. Just the acknowledgements for all the labs we got to work with, and also the Cure for Sanfilippo Foundation for actually funding this project to go to the next phase for our undergrads. And that is my sign. This last part was essentially just a public health sort of metric where I essentially did an estimate as to how much this actually costs society. So rare diseases are extremely rare. So it's difficult to convince policymakers, funding agencies, even average people, et cetera, that this is important. But for these individual families, the trauma can be quite profound. So the level of ethics, if you go to the highest, level of ethics, you theoretically have an egalitarian focus where you're focused on the suffering of individuals rather than the collectivist view, which is, just looking at how do you help the most amount of people, which is a bit more akin to sort of authoritarian regimes. So in America, like it or not, we tend to be very egalitarian with our philosophy. We care if one individual is suffering a lot. So with that logic, I sought out to essentially quantify how much trauma is caused from a health standpoint by having children with Sanfilippo syndrome in a world where we don't have any treatment for it. So I came up with a complicated algorithm for going over stage 1, 2, and 3 of the disease, Sanfilippo syndrome, for the patient and for the parents as well. What is the increased risk of having depression, anxiety, and PTSD? There actually have been studies on this, so I was able to get the numbers and validate them. And long story short, if we were to put value on the total amount of suffering that is happening because of this disease, it's valued at hundreds of millions of dollars per year. And to have one child with Sanfilippo syndrome can cost upwards of $6 million for the individual family over the lifetime of the child and the parents. And over the next 20 years, it's estimated to cost over $1.5 billion in present value. And I can answer any questions on that, obviously. Essentially, that's just to summarize all the work that we were able to do in the last 3 years. Any questions?
Clap, clap, clap. Well, I think you achieved your goal of making an engineer think he understood what you said. Thank you very much. So, any questions? Any Zoom questions?
No, but I have a question. Is this study ever look at diabetes in children? Or is it just focused on that one syndrome?
So in regards to gene therapy,
Yes.
So for diabetes type 1, we didn't really look at that specifically. So for type 1 diabetes, it's actually less about genetics. There's actually a very poor genetic correlation with diabetes. It tends to be actually acquired, and it involves the destruction of beta cells in your pancreas. So a lot of the therapeutics that are looking into diabetes type 1 are focused around suppressing the immune response to pancreas beta cells and regenerating the pancreas beta cells that were lost somehow.
Okay, thank you.
Yeah.
Thanks very much. How do you get a Sanfilippo mouse model? Are they going just for symptoms, or do they actually know that they have the same underlying genetic differences? And how do they make them?
Yes, thank you for that great question. So that was actually created in the 90s. So scientists have developed creative methods for altering the genome of the mouse. So we know the gene that deals with Sanfilippo syndrome. It's the NAGLU gene. So they can find that exact gene on the mouse genome, and they can cut it out, essentially. So those mice are essentially the same as genetically at least, they are the same theoretically as the Sanfilippo syndrome type B person. And those have been kind of bred for years, and they maintain them as a colony.
Well, we have a question here on Zoom. Rick Gold, go ahead.
Thanks very much. Can you identify a Sanfilippo during pregnancy? And what are the options for parents?
So that is a great question. Absolutely, you can. Do we do it? No. And the reason why is because from the perspective of healthcare as a resource, we can't just screen everyone for everything. Otherwise, we would go bankrupt after treating like 100 people, right? So there are newborn screening tools that we can use that are cost effective and also can provide prognostic value for parents and also help researchers to enroll in clinical trials so they can actually make progress on this disease. However, those are not being done. And that's one of the things that we've actually been trying to advocate for. There's a national organization called the RUSP, and they elect to the states make recommendations in terms of what diseases you can screen for and you should screen for. Because those are just genetic tests, and you can make those genetic tests cheap. So when each baby is born these days, they take a little bit of blood and they run that on a screen to see all of the genetic diseases, test to essentially see if the baby has any of them. But they don't do the ones where there's no therapy, and that's the problem. It's theoretically not ethical to screen for something if there's no treatment for it, is their reasoning. But the problem with that is then we can't identify those patients. And if we can't identify those patients, we can't enroll them in clinical trial. And by the time they're 8 and they get diagnosed, it's already too late because the disease has already progressed to the point where it's going to be very difficult to recover any function. I don't know if I answered your question, but that's the general premise of the situation here. So what we need to do is we need to advocate for more genetic screening for genetic diseases, even if there's no treatment for it, ideally, in a way that's cost effective and also can connect people with research trials that might potentially benefit future families.
Great, thanks.
Okay, I have a couple of things. I understand about putting the stem cells right into the brain for the blood-brain barrier, et cetera. But if the Sanfilippo, am I correct that it occurs in all the cells of the body?
That's correct.
Okay, and you know the gene?
Yeah.
Okay, so if you're going to work on now in the future fixing the gene, entering the correct gene in and getting it to the cells. How do you take, it's been a long time since I've done this kind of stuff. How do you take the correct gene, put it in the vector? And I know you have to work out how you get it in, but how do you actually get it into the vector?
Yeah, so that's a great question. So I intentionally left a lot of that out just because I didn't want to get in the weeds too much, but absolutely. So, right, exactly. So I appreciate that. So essentially the way that we make these AAV vectors is we have what's called a triple plasmid system. So we have plasmids that get put into human cells. These are HEK293 cells, which have been around since the 70s. We use them to essentially grow viruses, but we want to grow the viruses in a controlled way. I'm calling them viruses flippantly. They're not really viruses because what we're creating doesn't have any viral material in it, so it can't replicate on its own. AAV, even the wild type, can't replicate on its own. This one not only can't replicate on its own, it can't make a virus, period. It is just literally a vehicle for carrying our DNA, right? So the way that we make that is we have 3 different plasmids, one that encodes the actual capsid itself, one that encodes the DNA we want to put in the capsid, and the other is what's called a helper plasmid, because remember AAV, it requires another virus to help it out to replicate. So it needs to undergo that first replication cycle so it creates that vector that has our therapeutic gene. That vector can't replicate on its own. So what we have to do is very soon after we put these 3 plasmids into a bunch of cells, we have to purify them very soon and essentially do a lot of fancy stuff that takes multiple days and ensure that we get pure therapeutic vector. I don't know if that answers your question. Yeah, so the way that we would change what goes into the virus is we would modify those plasmids before we put them into the human cells. Yeah.
Does there seem to be any hereditary component of this syndrome?
Absolutely. So majority of cases do happen spontaneously. However, there are some established populations that do have this gene very prevalent in the population. So for those that don't know, you have 2 different copies of every chromosome, right? And so if you have one bad copy of this gene, you don't get any symptoms. So if there are a lot of people who just have no symptoms, and then they'll meet someone else who also is a carrier, and then those people have a 1 in 4 chance of having a baby with Sanfilippo syndrome, unfortunately. And Middle Eastern populations, European populations, they can be at a greater risk for this disease. Ashkenazi Jew is a very prevalent population for this disease and a lot of other lysosomal storage diseases. So if you're from an Ashkenazi Jewish background, it would be highly recommended to talk to a genetic counselor, before procreating to some degree, just so that you can anticipate and you can work out what the best options are. Because family planning is possible. And yeah, did I answer your question? Yeah.
Any other questions? Anything on Zoom? Fantastic. Thank you so much.